RNA structure. Like DNA, RNA is composed of nucleotides with a sugar phosphate backbone and 4 unique nucleotides. There are differences. RNA's sugar is ribose. RNA is a single chain of nucleotides and has multiple shapes. DNA is always a double helix. Instead of thymine, RNA has uracil, U. RNA brings the genetic code stored in DNA out of the nucleus and into the cytoplasm, where the machinery of the cell uses the information to create proteins. RNA's 4 bases bond in complementary pairs, with uracil replacing thymine. Adenine A bonds with uracil, U. Guanine G bonds with cytosine, C. U requires less energy to produce, but is not as stable as T. T is better for long-term storage of genetic information, which makes it ideal for DNA. U is more efficient, but exists for a shorter period of time, which makes it more useful for RNA. RNA molecules fold and have higher order levels of structure stabilized by hydrogen bonds. As in proteins, the shapes of RNA molecules help to determine their functions. So there are primary, secondary, tertiary, and quaternary structures to RNA that help determine their functions. A strand with bases in the sequence ACU, AGG, UAC, AUG has a different shape and functions differently than a strand with the sequence CUA, GAA, CAG, UCAA. Letter changes result in a new shape and new functions. These are illustrative sequences and images of different RNA shapes. 3 main types of RNA are required to take the DNA genetic message from the nucleus to the cytoplasm to build proteins. Many other types of small regulatory RNAs are also involved in gene expression but are beyond the scope of this course. Messenger RNA, or mRNA, carries the DNA genetic message from the nucleus to the cytoplasm. Ribosomal RNA, rRNA, along with additional proteins, make up the ribosome. The ribosome catalyzes the reaction that builds proteins from amino acids. Transfer RNA, tRNA, carries amino acids to the ribosome, which will covalently bond them together to form a protein. Their functions will be described in detail in the next unit. 3 main types of RNA. Each type of RNA has a different shape, which leads to their different functions. RNA strands fold in on themselves, forming hydrogen bonds between nitrogenous bases and between the bases and the surrounding water. These bonds cause RNA molecules to form different shapes. A nucleotide with an adenine base will hydrogen bond with a nucleotide with a uracil base, and a nucleotide with a guanine base bonds with a nucleotide with a cytosine base. Originally, RNA played many roles that have now been taken over by more specific molecules. RNA's critical function that remains is the creation of proteins. RNA at one time catalyzed reactions, that's now done by enzymes. Also at one time transformed energy, that's now done by the mitochondria. Before it was the storage for genetic information, that's now done by DNA. DNA versus RNA. Its double helix makes DNA a better archive for genetic information since the bases are protected inside of the helix. Also, thymine is more stable than uracil. But its helical structure means that DNA can't directly work in the cell. It is a library of information, but the only way that information can be used is via RNA. RNA is chemically active in the cell, DNA is not. DNA is useful and stable as an archive. It exists for generations and has been modified over billions of years. RNA has a half-life of minutes. DNA is a long-term storage of the entire genetic code for the organism, whereas RNA is used to transfer segments of code or genes to the ribosomes. DNA is stable under extreme conditions such as high and low temperatures, pH extremes, high and low salt environments, etc. It is easy to work with. RNA is not stable and is hard to work with. RNA is useful working in the cell. DNA is maintained in a safe environment to maintain the integrity of the genetic information. RNA is used throughout the cell to implement the genetic information that is stored within the DNA.